Hello, everyone. So this is part 2 of the genetics basics presentation. And originally I had thought that it would be all done in 2 parts, but then in looking at the amount of material that I have to cover, it's actually going to be 3 parts. So this is going to be part 2 of 3, which is titled The Types of Genetic Mutations. So if you remember from the previous talk, what we did is to make an analogy of a letter of a sentence composed completely of 3-letter words to illustrate the way in which a gene codes the amino acid sequence of a protein, because every 3 DNA letters correspond to a particular amino acid. So the base sentence without any changes or mistakes is the cat saw the rat but not the bat, and then it ends with an out, which is our code word for the end of the sentence. Okay, so for the first type of mutation that I'm going to describe is what's called a nonsense mutation. And it turns out in genetics, it has a completely different meaning than what you probably think nonsense means. You know, you normally think of it as just it's something that has no meaning whatsoever. But in genetics, it has a very precise definition. And what that means is that you stop creating the amino acid sequence of the protein before the real end of it. So our code word in this sentence analogy is the word out. That signals the end of the protein. So an example of the nonsense mutation, if you change the B in the original sentence in the word but, which is about halfway through, the cat saw the rat but, if you change the B to an O, then it reads the cat saw the rat out. Okay, but out means the end of the sentence. So the cell in making the protein is going to ignore the rest of the sentence. So it knows that the cat saw the rat, but doesn't know anything about the bat. So the protein is shortened. Okay, so an example in a real DNA, and the way that this is specified in a genetics report. Okay, so the way that mutations are stated in a genetics report, they're in 2 parts. The first is with a C abbreviation. And what that means is what happens at the DNA level. And the second part is begins with a P, and that says what happens with the amino acid sequence of the protein. Okay, so in this example, for a nonsense mutation, you change the base pair number 6086 in the DNA sequence of a certain gene from a C to a T. Okay, that doesn't really tell you very much. But in that particular sequence, what that does is it changes the amino acid number 1905 from the amino acid arginine, which is abbreviated with an R, to a stop codon, which is abbreviated with an X. So an X means that that's the end of the protein. So basically anything after that point of the protein isn't going to be there. And if that's necessary for the protein to work, then this mutant protein won't work. Okay, the second type of mutation is called a missense mutation. And what this means is you're changing one amino acid to another. In our sentence analogy, we're changing one word to another. Okay, so the original sentence was the cat saw the rat but not the bat, but now we change the R to an H, and so that the cat saw the hat but not the bat. Well, a bat and a hat are pretty different things, so that might change the meaning of the sentence. Okay, so an example of this is we have in our protein, which it turns out is actually the dysferlin protein, we change a C to a T, which was actually the same DNA base change we had in the last example. But this time, instead of changing from an R again to an X, we're changing to a W, which is a different amino acid. So this doesn't end the translation of the gene into a protein, but it has the wrong amino acid in a certain place. And in many cases, even though you have hundreds or thousands of amino acids in a protein, having just one incorrect can cause the protein not to function correctly. Okay, the third type of mutation that I'll talk about is what's called an insertion. And that means you just stick different additional DNA bases in the gene that aren't supposed to be there. Now here we stick the word Bob, although it turns out that it's split up between different three-letter words, in other words, codons. So you have a couple of three-letter words that don't really make any sense. Okay, so an example of that, in this case, we are inserting three additional DNA letters, which means usually that you just put in one extra amino acid that's not supposed to be there, or sometimes you change one amino acid to two different ones that aren't really what they're supposed to be or aren't supposed to be there. So here we put in three bases, CGC, and then add in one additional amino acid, and R, in between, and then the genetic notation says, the two amino acids that the extra one is stuck in between. Now, the next type of mutation is what's called a frameshift. And this is actually a very common type of mutation. It's when you either put in one or more DNA bases or take away one or more DNA bases, and the number that you put in or take away isn't a multiple of 3. Remember that, unlike the sentence analogy, there's not really any spacing between words. Every, 3 DNA letters is assumed to be a new word. So if you put in just one additional DNA base, kind of in the sentence analogy, you get a bunch of gibberish that isn't even words. Now, in DNA, it turns out every possible 3-letter sequence is actually a word. However, every amino acid that it specifies is going to be completely wrong. So it's very unlikely that you're going to get anything that works at all. And we see that our happy little cloud is not at all happy. Okay, so for real DNA, so at base 3505, we stick an extra DNA letter C in, which then changes the L to a Q at amino acid number 1169, and then causes a stop codon to occur several amino acids downstream. And that's typically when you get a frame shift, you end up with a stop codon pretty quickly, just because if you just had random DNA letters, about 1 of every 20 codons would be a stop. Okay, so a special kind of insertion is called a duplication, and that is that you just, repeat 1 or more letters of DNA. And so that really works pretty much the same way. It can either be in frame or out of frame, meaning in frame means that the number of letters you insert is a multiple of 3. Out of frame means that it's not a multiple of 3. And the real DNA example, we're just duplicating an A in a certain position, which again causes a frame shift, changes the amino acid, and then causes a downstream stop codon. Now, opposite an insertion, instead of putting extra DNA letters in, we can take them away where they're supposed to be there. So our sentence example is, we just leave out the word rat. The cat saw but not the bat. Okay, well, that doesn't really make any sense. The cat saw the what? And, that's because there's a missing word. So the way that a deletion is reported in a DNA analysis is shown at the bottom. And this example, we have the deletion of 15 base pairs, and this would be starting with base number 598 through 612. Normally, if only 1 or 2 bases are deleted, you actually would write out what the bases are, like del A or del AC. But in this case, there's so many of them, it's kind of unwieldy to write out, so you just say del 15. And since 15 is a multiple of 3, that means that there's going to be 5 amino acids which are missing from the final protein. And so on the protein portion, you're missing amino acids 200, which is an F, through 204, which is an L. So this is the end of part 2. In part 3, we'll go over the way that genes are spliced together to make the final sequence for a protein. And there's a number of types of mutations which impact splicing. So we have to talk a little bit about splicing to explain how those work. So thank you again for watching.